Let's dive into the liquid matrix, starting with Section 1, the liquid lifeline blood composition and physical properties.
Right. First thing, we need to set aside the simple visual of blood as just a red liquid. Physically and chemically, it's a highly engineered fluid. It's optimized for carrying mass and maintaining pressure.
And the sources describe it as an opaque fluid. That opacity, that tells you something immediately, doesn't it?
It tells you it's packed. It's packed with suspended particles, cells, fats. It's not just a clear liquid with dissolved salts like, salt water. It's a suspension.
I noticed some very specific, really clinical level measurements in the sources. The relative viscosity is listed as about 4.75 at 18 degrees Celsius, and its specific gravity is 1.06 at 15 degrees Celsius. Yeah. For the average person, what do those numbers really tell us about the fluid we're pumping around our bodies?
Okay, so they tell us two absolutely critical things. First, the viscosity of 4.75. That means blood is nearly 5 times thicker, 5 times more viscous than pure water.
5 times? That's a lot.
It's a huge amount. And that density, that thickness, is essential. It contributes significantly to something called vascular resistance, which is just a fancy way of saying it helps maintain the blood pressure necessary to circulate it across, the vast distances in the body.
So if our blood were as thin as water, we'd have a serious problem.
A massive problem. We'd struggle hugely to maintain systemic pressure. The heart would have to work in a completely different and probably impossible way.
So it's a thick, highly substantial carrying fluid, not just a simple vehicle. And the color is also a constant indicator of its function, isn't it?
Absolutely. The color difference reveals the functional state of the fluid at any given moment. The bright scarlet red we all associate with, say, a cut artery that's oxygenated arterial blood. It signifies high oxygen saturation, ready to fuel tissues.
And the other side of that.
In contrast, systemic venous blood, which is on its way back to the lungs after offloading its payload, appears a much darker sort of purple red. This visual contrast is a fundamental indicator of metabolic activity. It's showing you oxygen exchange efficiency across the whole body in real time.
Okay, this is where it gets really interesting for me. Flow dynamics. The sources make a specific point that blood flow is not entirely predictable by simple Newtonian equations.
Right.
Why is our blood non-Newtonian? That sounds like something out of a physics textbook.
Well, it's because of its multi-component nature. So Newtonian fluids, like water, have a viscosity that stays constant no matter what stress you apply to it. Water is always just as thick. Blood doesn't do that.
Why not?
Because it's simultaneously a true aqueous solution of molecules, salts and proteins dissolved in plasma, and it's a suspension of billions and billions of cells, the erythrocytes, leukocytes, and platelets. It's two things at once.
So when it flows quickly through a large artery, it behaves differently than when it's squeezing slowly through a tiny capillary.
Precisely. In large vessels, the central column of cells actually flows faster, and near the vessel walls, the flow is slower. It creates a kind of shear. But when it enters those tiny capillaries, which can be narrower than the cells themselves, the cells have to deform in a line.
They line up, single file.
They do, and they fold over.
Yeah.
And this completely changes the fluid dynamics at that microscopic level. This complexity is why studying microcirculation is so difficult. But that dynamic adaptability is absolutely vital for efficient tissue perfusion.
So let's talk about the liquid solvent component of this complex fluid, plasma. This is the clear, straw-colored liquid that makes up the largest volume of blood. I know it's mostly water, about 92%. But what's in that small remaining percentage, and how is it all managed?
The water content is incredibly highly regulated, and you can see that regulation reflected in its freezing point depression, which is consistently maintained at minus 0.54 degrees Celsius.
That seems like a very specific number to maintain.
It is, because it's a critical osmotic constant. Any variation in that number is a huge red flag for a clinician because it reflects the total concentration of all the dissolved particles, the small ions and the large macromolecules. It's a measure of total solute concentration.
And the ions themselves are, well, they're the engine of communication and balance. We're talking about the key electrolytes here. Sodium, chloride, potassium, calcium, magnesium, phosphate, bicarbonate ions. Why is maintaining the exact concentration of these specific ions so crucial?
Because they do everything. They maintain the body's osmotic balance, which stops cells from shrinking or swelling up like balloons. And they are absolutely essential for action potentials.
Nerve impulse.
Nerve impulses, muscle contraction. Sodium and potassium, they are the foundation of the entire nervous system. Calcium isn't just for bones. It's a massive signaling molecule and a vital, absolutely vital cofactor for blood clotting. These concentrations are constantly being monitored and adjusted by the kidneys and various endocrine glands.
Beyond the small ions, we move into the heavy hitters, the high molecular weight substances, the macromolecules. These are the proteins and lipids that really define plasma's functional power.
Yeah, and we can basically categorize these essential macromolecules into three major functional groups. First, you have the blood clotting factors, things like prothrombin and fibrinogen, which are just circulating, waiting for the signal to initiate a repair cascade.
The emergency repair crew.
Exactly. Second, you have the immunological defense proteins, which are the immunoglobulins. These are your circulating antibodies, the long-range weapons of the immune system. And third, you have a huge variety of transport proteins.
Like what?
Well, lipoproteins, which are designed to carry fats because fats and water don't mix. And then globulins, which transport all sorts of things, metals like iron and copper, hormones, and other necessary small molecules that can't just float around on their own.
This detail really highlights why measuring plasma components, the concentrations of enzymes, electrolytes, proteins, and so on, is the absolute foundation of modern clinical diagnosis.
It's incredible. What's truly fascinating is that the simple act of drawing a tube of blood gives you an immediate chemical snapshot of the entire body's health. If you see high levels of certain liver enzymes in the plasma, right away, there is cell damage happening in the liver.
Because those enzymes have leaked out of the damaged cells into the bloodstream.
Precisely. If the protein balance is off, you might suspect nutritional deficiencies or serious kidney disease. The plasma is a constantly updated report card on the health of every major organ system, and that includes a crucial reflection of endocrine, hormone function.
But that detailed analysis depends entirely on how you handle the blood in the lab. We have to discuss how blood is prepared because that very first step determines what you can ultimately study.
Exactly right. When blood is just drawn and left untreated in a tube, it separates into two things, a solid clot and a liquid called serum. And serum is what's left of the plasma after all the clotting factors like fibrinogen have been used up and removed in the clot.
So if you want to study the clotting factors themselves, you have to stop that process from ever happening.
You must prevent it entirely. And that's where anticoagulants come in.
You mentioned a couple of common ones that interrupt the cascade.
Yeah, and the choice of which one to use depends on the mechanism you want to target. A very common one is EDTA, which stands for ethylene G-acetic acid. It's commonly used because it chelates, which is a chemical term for grabbing onto and binding calcium ions. It effectively removes all the free calcium from the sample.
And since calcium is a non-negotiable cofactor for almost every single step in the clotting cascade,
removing it stops the process dead in its tracks. No calcium, no clot.
So if you were to accidentally use a tube with EDTA for a test that required the blood to clot, the test would just fail completely.
It would fail because you've removed the mineral spark plug, so to speak. Then we have another common one, heparin. Heparin acts differently. It doesn't touch the calcium. Instead, it works by massively enhancing the activity of a natural inhibitor called antithrombin.
So it boosts the body's own off switch for clotting.
That's a great way to put it. It boosts the natural brakes. So, depending on the research or clinical test, the specific mechanism of anticoagulation is chosen very deliberately.
I also saw mention of defibrinating agents. That seems counterintuitive. You're trying to prevent clotting, but you use something that actually forms a clot.
Yeah, that's used for very specific studies where you need the plasma solution, but you need to be absolutely certain there's no active fibrin or fibrinogen floating around that could interfere with your measurement.
So you force a clot to form just so you can remove it.
Exactly. You intentionally initiate fibrin clot formation, usually by stirring the blood with a glass rod or adding glass beads, and then you physically wind up and remove the resulting fiber strands. You're left with a specific solution that's often necessary for certain cellular or immunological analyses.
It's incredible how much chemistry is involved just in getting a clean sample.
The preparation is foundational to any reliable analysis. Garbage in, garbage out.
Okay, now we transition from the liquid matrix to the most populous cells it carries, the erythrocytes, or red blood cells. This is the red brigade, the transportation system designed for one singular critical purpose.
And they're present in just staggering numbers. This dictates the massive scale of the body's logistics. We're talking 4.1 to 5.1 trillion cells per liter in females and 4.7 to 5.5 trillion cells per liter in males.
Trillion. It's a number that's hard to even comprehend.
It is. And the difference between males and females might seem minor, but it reflects real physiological distinctions, like hormonal status and average muscle mass.
So any shift in these astronomical counts, polycythemia, meaning too many, or anemia. Too few immediately signals a system under serious stress, and it all begins with the incredible design of the individual cell.
The erythrocyte structure is an evolutionary marvel. It's a characteristic biconcave, disc-like, a donut that's been pressed inward at the center, but without the hole going all the way through.
And the dimensions are tiny, about 7.8 micrometers in diameter.
Yes, but the crucial engineering is in its shape, that thin center and a thicker rim.
Why that specific shape? Wouldn't a simple sphere be, easier for the body to produce?
A sphere would maximize volume, but it would minimize the surface area for that volume. The biconcave shape provides an exceptionally high surface area to volume ratio.
Which means more surface for gas to move across.
Exactly. It dramatically enhances the rate of gas exchange oxygen loading in the lungs and offloading in the tissues compared to a spherical cell of the same volume. Furthermore, they are essentially just bags of hemoglobin. They've deliberately ejected their nucleus and most other organelles during maturation to maximize space for hemoglobin.
And that lack of internal machinery means they are incredibly flexible, which ties right back to the non-Newtonian flow we discussed earlier.
Absolutely. They are constantly deforming. When they slow down in the small veins, the venules, they align face-to-face like stacks of coins. It's a process called rouleau formation.
Like a stack of poker chips.
It looks exactly like that under a microscope, but when they enter a capillary that is sometimes narrower than their own 7.8 micrometer diameter, they have to fold over, becoming paraboloid, and just squeeze through one by one.
Without that immense flexibility, we'd have continuous capillary blockages, a constant state of microstrokes.
We would. The system would grind to a halt.
But how does a cell without a nucleus, constantly being squeezed and sheared, maintain that specific biconcave shape for 120 days? It sounds like they would just burst.
That's the structural genius we need to unpack. The plasma membrane has the typical lipid bilayer, sure, but underneath that, acting like an internal suit of flexible armor, is a dense protein cytoskeleton.
And what are the key elements of that armor?
Okay, so you have transmembrane proteins like glycophorins and a protein called band3, which anchor the whole structure to the outer membrane. But the real strength comes from the internal scaffolding, and that is primarily composed of a protein called spectrin.
Spectrin.
Spectrin is a fibrous protein that forms a mesh-like network, and it's linked back to the membrane via accessory proteins like ankyrin and another called band 4.1 protein. And you also have structural elements like actin and tropomyosin in there as well.
So it's like a geodesic dome built just beneath the surface, allowing for maximum flexibility while resisting the constant shear stress of circulation.
That is a perfect analogy. And the clinical relevance of this is profound. If there are inherited defects in this spectrin ankyrin network, the cell can't maintain its biconcave shape. It becomes fragile, less deformable, and often more spherical.
Which is a problem for the spleen.
A huge problem. The spleen acts as the body's quality control filter, and it recognizes this abnormal shape. It pulls these fragile cells out of circulation and destroys them prematurely. This leads to a whole class of diseases called hemolytic anemias, like hereditary spherocytosis or elliptocytosis. The microscopic structure dictates the entire pathology.
And inside that perfect package is the machinery for transport, hemoglobin, or HB. This is truly where chemistry meets life.
Hemoglobin is a massive globular protein, about 67 kilodaltons, and it's built to interact with oxygen in a very specific way. In adults, the main form, HbA, consists of 4 globin molecules, 2 alpha chains and 2 beta chains. And nestled within each of those 4 chains is a non-protein structure called the heme group.
And at the absolute heart of that heme group is a single critical atom of iron.
That's right, 1 iron atom per heme group. So each complete HB molecule holds 4 iron atoms, and therefore each iron atom can bind 1 molecule of oxygen. But the binding process itself is highly sophisticated. It's known as cooperative binding.
Explain that cooperation. It sounds like one oxygen molecule binding somehow influences the others.
It does, in a big way. When the very first oxygen molecule binds to one of the 4 sites, it induces a physical conformational change in the entire protein structure. It twists it slightly. This change shifts the remaining 3 sites from a low-affinity tense state, or T state, to a high-affinity relaxed state, or R state.
So binding the first one makes it easier for the others to bind.
Dramatically easier. This makes it much, much easier for the 2nd, 3rd, and 4th oxygen molecules to bind rapidly.
That's ingenious. It means that when you're in the lungs where there's a high oxygen environment, the hemoglobin loads up with oxygen extremely quickly and efficiently.
And the reverse is true in the tissues, where there's a low oxygen environment. As soon as the first oxygen is released, it shifts the protein back toward the tense state, making it easier for the other 3 to drop off their cargo. This cooperative switch is fundamental to efficient oxygen delivery. If binding were just linear, one for one, we would suffer systemic hypoxia.
This incredible machine has a limited lifespan, though, about 100 to 120 days. When we talk about logistics, we have to talk about the sheer scale of the turnover required to maintain this system.
The scale is almost incomprehensible. To maintain stable counts, the body must destroy and replace approximately 6 million red cells every second.
Every second.
Every second of every day. So every 4 months, you essentially have a brand new supply of oxygen carriers. This constant non-negotiable turnover places an incredible demand on the bone marrow factory.
Once they hit that 120-day mark, the cells become more fragile, their surface proteins change, and they are recognized by the body as obsolete. Where does the recycling happen?
Primarily in the mononuclear phagocyte system. Specifically, in the macrophages that reside in the spleen, the liver, and the bone marrow itself. They phagocytize or eat the aged cells and begin this very precise multi-step breakdown process.
The globin chains are easily dealt with. I assume they're just protein.
Yeah, they're hydrolyzed back into their constituent amino acids, and those are just recycled into new proteins. But the heme group requires very careful handling. It's potentially toxic.
So how is it detoxified?
The heme ring, this porphyrin ring structure, is opened up. It's converted first to a green pigment called biliverdin, which is what gives old bruises their green color. Then that's rapidly converted to bilirubin.
And bilirubin is the problem if it builds up.
It is. It's toxic if it's allowed to circulate freely, so it's immediately bound to the plasma protein albumin for transport.
And then it's shuttled to the liver. This is the crucial step related to conditions like jaundice, correct?
Exactly. The liver performs a process called conjugation. It chemically modifies the bilirubin to make it water-soluble. Then it can be excreted via bile into the digestive tract. If there is liver dysfunction or a blockage, or if red cell destruction, hemolysis is happening too quickly for the liver to keep up, unconjugated bilirubin builds up in the blood.
Leading to the yellowing of the skin and eyes that we recognize as jaundice.
Precisely.
And the iron atom. That single precious atom that can't be wasted in a system this efficient.
It is not wasted. It's liberated from the heme group and immediately bound to a transport protein called transferrin. It's either shuttled straight back to the bone marrow for immediate new hemoglobin synthesis, or it's taken to be stored in the liver and spleen, primarily in large molecular complexes called ferritin or hemosiderin. It's a completely closed-loop system designed for maximum recovery of this essential mineral.
Before we move on, we have to touch briefly on blood groups because they demonstrate the crucial immunological identity of these cells. We all know ABO and RH, but the source material hints at a much, much wider complexity.
Oh, it's hugely complex. The source of antigenicity, what makes blood types different, comes from specific glycolipids and glycoproteins that decorate the red cell surface. While we won't list all 20-plus systems like MNS, KEL, Duffy, or KID, the conceptual takeaway is vital.
Which is?
Each of these systems represents a set of tiny molecular flags that the immune system uses to identify self.
So when a transfusion reaction occurs, it's because the recipient's immune system recognizes a molecular ID tag on the donor's red cells, say a protein from the Duffy system, as foreign, triggering a potentially catastrophic immune response.
It's a matter of life or death, and it's dictated by microscopic sugars and proteins on a cell surface. This is why blood typing and cross-matching is such a complex immunological endeavor, even though ABO and RH incompatibility are by far the most common and severe risks.
Shifting now from the oxygen carriers to the body's repair and maintenance crew, let's look at platelets, or thrombocytes, the clotting masters.
And these are not cells in the traditional sense. They are tiny, non-nucleated fragments, only about 2 to 4 micrometers in size. And they are derived from the cytoplasm of the absolutely massive megakaryocytes in the bone marrow.
And the normal count, 200,000 to 400,000 per microliter, shows they are numerous, but they're completely dwarfed by the red blood cell count.
Completely. But their structure is perfectly organized for explosive activation and mechanical action. They possess a very robust actin-myosin cytoskeleton, which is ready for contraction, and their surface membrane is studded with critical adhesion proteins.
What are the most important of these surface proteins?
Well, there's glycoprotein IDXV, which helps them initially adhere to damaged vessels. But the real key player is a different one, glycoprotein IBDA. This acts as the binding site for fibrinogen once the platelet becomes activated.
And internally, they're like tiny warehouses stocked with signaling molecules and structural components, all stored in two main types of granules.
That's right. The dense bodies contain the small molecules, ATP ADP, which is the primary platelet activator serotonin, also known as 5-HT, and essential calcium ions. These are the immediate release signaling components that get the ball rolling.
And the alpha granules hold the larger molecular weight components, the structural elements.
Correct. Things like clotting factors, massive amounts of fibrinogen, platelet-derived growth factor or PDGF for later tissue repair, thrombospondin, and von Willebrand factor, or VWF, which is absolutely crucial for that initial adhesion step.
So they function in hemostasis. The cessation of bleeding is a rapid, finely tuned cascade.
It is. Upon injury, the vessel wall is breached, and it exposes subendothelial collagen. Platelets then adhere to that exposed collagen via VWF and their IBXV receptors.
And that's the trigger.
That's the trigger. Activation then occurs, causing the release of all the contents from the granules. The ADP that's released activates surrounding platelets, causing a conformational change in their IBA receptors, which basically makes them incredibly sticky.
And that stickiness is what forms the temporary plug. The IBA receptors grab onto fibrinogen, linking multiple platelets together into a cohesive mass.
Right. And at the same time, the serotonin that's released promotes local vasoconstriction to try and minimize blood loss from the outset.
Once that initial plug is formed, the next phase is clot retraction, which you said is entirely mechanical.
It is. The actin-myosin cytoskeleton inside all those activated platelets begins to contract, like millions of tiny muscles. They pull on the newly formed fibrin network, which tightens the whole clot and actually pulls the edges of the damaged vessel together, like a microscopic purse string being cinched tight.
And since a clot can't stay forever, the system needs a way to dissolve the old repair job once the vessel wall has healed.
And that process is called fibrinolysis. Platelets themselves are very short-lived, with a half-life of only about 10 days. But the long-term removal of that fibrin mesh is accomplished by enzymes, primarily one called plasmin, which systematically degrades the fibrin network, clearing the vessel for normal flow again.
It's an elegant system of build, contract, and then degrade.
Perfectly balanced.
We've covered transportation and repair. Now let's shift gears entirely and look at the critical part of the blood that isn't about flow, but about the fight. The leukocytes, or white blood cells, the body's highly specialized defense system.
And these cells are far, far less numerous than red blood cells.
Mm-hmm.
We're talking typically circulating between 4.0 and 10.0 billion per liter. But what they lack in quantity, they more than make up for in functional specialization.
We categorize them into 5 major classes based on their structure, specifically whether they contain granules and how their nucleus is shaped.
That's right. We have the granulocytes, neutrophils, eosinophils, and basophils, which have visible granules and these irregularly lobed nuclei. And then the granulocytes, monocytes, and lymphocytes, which have simpler, non-lobed nuclei.
Let's start with the largest group and the absolute front line, neutrophils. They're also known as polymorphonuclear leukocytes because of that multi-lobed nucleus.
And they represent 50 to 70% of the entire white blood cell population. They are the foot soldiers.
They are about 12 to 15 micrometers, and that multi-lobed nucleus, 3 to 5 distinct lobes connected by fine threads, is their absolute signature. But the real arsenal is in those faintly staining granules.
Yes, and we have to differentiate between their different weapons depots. The primary, or eosinophilic granules, are essentially powerful lysosomes. They are formed early in the cell's development.
Then they contain
potent killing enzymes like myeloperoxidase and acid hydrolases.
Okay.
And myeloperoxidase is critical because it reacts with hydrogen peroxide and chloride ions to produce hypochlorous acid.
Which is?
It's essentially bleach, right inside a phagocytic vacuole in the cell.
Wait, they create bleach internally? That's an extreme measure for internal defense.
It is the ultimate microbial acid bath.
Mm-hmm.
Then later in their development, they form the secondary or specific granules, which are much more numerous. These contain things like lactoferrin, which binds iron and basically starves bacteria, and collagenase and lysozyme, which break down bacterial cell walls and surrounding connective tissue to help them move.
Their primary function is rapid response, phagocytizing bacteria and cellular debris, driving acute inflammation. How exactly does this respiratory burst killing mechanism work in practice?
Okay, so the oxygen-dependent mechanism is highly energy-intensive. Once the neutrophil engulfs a microbe into a phagocytic vacuole, a complex of enzymes rapidly consumes oxygen, that's the respiratory burst, to generate massive quantities of reactive oxygen species, or ROS.
Things like superoxide and hydrogen peroxide.
Exactly. And then that hypochlorous acid we mentioned, this rapid targeted oxidative stress just neutralizes the microbe almost instantly.
But if they are deployed in an area of severe trauma or infection, oxygen might be scarce. Do they have a backup plan?
They do. That's the oxygen-independent killing mechanism. They just flood the phagocytic vacuole with the acid hydrolases and myeloperoxidase from those primary granules, digesting the target using pure hydrolytic enzymes, regardless of the ambient oxygen levels.
And the sources mention a relatively recent discovery in defense, the formation of nets. What are neutrophil extracellular traps?
This is fascinating. When faced with certain pathogens, the neutrophil can initiate a unique programmed cell death pathway called NEETosis. It essentially sacrifices itself by expelling a massive, sticky web composed of its own decondensed chromatin, its DNA, which is laced with all those toxic granule enzymes.
So it throws out a sticky poison net.
It acts like biological flypaper. It traps and immobilizes bacteria outside the cell, preventing their further spread until the big cleanup crew, the macrophages, can arrive.
Their commitment is intense, but brief. They circulate for only 6 to 10 hours before migrating into the tissues, where they survive for only a few days unless they're consumed in the fight.
Very short-lived. Next, we have eosinophils, which make up about 1 to 5% of the total white blood cells. They're easily spotted by their usually bilobed nucleus and their highly distinct, bright, pinkish-red granules that stain very strongly with the dye eosin.
And their arsenal is specialized. What is the major basic protein found in their granules?
Major basic protein, or MBP, is highly toxic. Eosinophils specialize in tackling large parasites, particularly Hellman's worms, which are way too big to be engulfed by a single phagocyte.
So they can't eat it. What do?
Instead, the eosinophil degranulates directly onto the parasite surface. It just dumps its toxic cargo using MBP and other proteins like eosinophil cationic protein and neurotoxin to punch holes in the parasite's outer membrane, effectively executing it externally.
And we also associate eosinophils heavily with allergic responses.
Yes, they are strongly chemotactic, meaning they are chemically attracted to sites of inflammation and allergy. They actually help to dampen the allergic response by releasing enzymes like aryl sulfatase and peroxidase, which inactivate the potent inflammatory mediators like histamine and leukotrienes that are released by other cells.
So they're the body's attempt to put the allergic fire out.
Exactly. They come in after the initial explosion to try and clean up.
The third granulocyte, the basophils, are the ghost cells, less than 0.5% of the total. Their irregular nucleus is often completely obscured by these massive, deep blue staining granules.
And those granules are like little chemical time bombs. They are rich in heparin, which is a natural anticoagulant, histamine, a major vasodilator that increases blood flow and capillary permeability, allowing other immune cells to flood into the tissue, and leukotrienes, which promote smooth muscle contraction, something that's particularly relevant in asthma.
So their function is tied directly to triggering immediate hypersensitivity.
That's their main job. Basophils and their tissue counterparts, which are called mast cells, possess high-affinity surface receptors for a type of antibody called IgE.
Okay.
When an individual is sensitized to an allergen, say pollen, their body makes IgE antibodies that then stick all over the surface of these cells. Upon a subsequent exposure, the allergen binds directly to that IgE, and that instantly triggers a massive degranulation, the explosive release of all those mediators.
And that drives the symptoms of acute allergies and asthma, the runny nose, the wheezing.
All of it.
The distinction between the circulating basophil and the tissue-resident mast cell is important.
It is. Mast cells are fixed in connective tissues throughout the body, and they're key players, not just in immediate hypersensitivity, but also in chronic inflammatory diseases like rheumatoid arthritis. Their ability to release these powerful mediators makes them a major focus for developing new anti-inflammatory therapeutic targets.
Let's move to the agranulocytes, starting with the mononuclear phagocyte system, which originates with monocytes. These are the largest leukocytes, 15 to 20 micrometers, and they're characterized by that eccentric kidney-shaped nucleus.
Monocytes are highly motile, but they only circulate briefly, maybe for a few days. They are essentially precursors or trainees. Their true destiny is in the tissue, where they migrate and then differentiate into tissue-specific, long-lived macrophages.
And the macrophages are the versatile veterans. They are huge, up to 25 micrometers, and they are tissue-specific, each with its own local name.
Right. Kupffer cells in the liver, alveolar macrophages in the lungs, osteoclasts that resorb bone, and microglia in the central nervous system. All macrophages, just with different specialties.
Structurally, they are built for industrial-scale cleanup, with abundant, rough endoplasmic reticulum, Golgi, and numerous vacuoles for digesting material.
Their functions are immense, They clear debris, they ingest dead and dying cells, they scavenge foreign material, and they provide constant immune surveillance. They are also crucial cytokine secretors, releasing factors like interleukin-1, IL-1, and tumor necrosis factor alpha, TNF-alpha, which basically coordinate the entire inflammatory response.
This brings us to the core mechanism shared by neutrophils and macrophages. Phagocytosis, the act of engulfing particulate matter. How does the cell decide what to engulf?
It starts with highly specific recognition. Phagocytes are covered in what are called pattern recognition receptors, or PRRs. A major family of these are the toll-like receptors, or TLRs.
And what do they recognize?
They recognize broad, conserved molecular patterns that are common to many pathogens, but not found in human cells, things like bacterial cell wall components. They also have receptors for complement proteins, which are proteins that the body uses to tag targets for destruction. It's called opsonization.
So the body uses a combination of general warning signs from the TLRs and specific eat me flags from the complement receptors to identify targets.
Exactly. And while neutrophils are geared towards the rapid destruction of small bacterial targets, macrophages are the primary consumers of large debris, old cells like those aged erythrocytes we talked about, and they are absolutely central to maintaining tissue homeostasis.
And this cleanup rule elevates these cells to the status of antigen-presenting cells, or APCs. This is the crucial functional bridge between the rapid, nonspecific innate immune system and the highly specific adaptive immune system.
It is the most important link. APCs, and the main ones are macrophages, dendritic cells, and B cells, don't just destroy what they ingest. They process the debris, they break down the foreign proteins into small fragments, and then they display those fragments on their surface using specialized molecules called MHC class 1 or class 2.
This display is essentially a highly organized intelligence briefing. It's shouting to the T lymphocytes, look what I found, this is a foreign threat. You need to figure out a specific long-term response to this.
And if we're going to talk about intelligence, the absolute elite of this class are the dendritic cells, or DCs. They are the ultimate immune sentinels. They're found in the blood, lymph, and strategically located throughout mucosal tissues and the skin.
The best studied example are the Langerhans cells in the epidermis. What makes DCs the most potent APCs?
They are specialized for two functions. First, capturing the antigen. They are constantly sampling their environment. Second, and this is crucial, they are specialized for migration. Once a DC captures an antigen in the skin or some other periphery, it dramatically changes its function. It stops capturing, leaves the tissue, migrates up the lymphatics, and travels directly to the secondary lymphoid organs, the lymph nodes.
To do what?
To find and prime naive T cells. They are the essential initiators of the adaptive immune response. They deliver the intelligence directly to headquarters.
If the White Army is the field force, then the lymphoid tissues are the strategic command centers and training facilities. This is where the decisions for long-term immunity are made.
And we classify these centers into primary and secondary tissues. The primary tissues, the bone marrow and the thymus, are the birthplaces and the training grounds where lymphocytes are born and undergo their rigorous maturation.
And the secondary tissues, the lymph nodes, the spleen, and multi, are the patrol zones, the meeting points where the mature lymphocytes encounter foreign antigens and launch a response.
Let's focus on the lymph nodes for a moment. These are small, encapsulated kidney-shaped organs, and they're strategically placed all along the body's major lymphatic vessels. They filter the lymph before it gets returned to the blood.
Structurally, they are architectural masterpieces. They're organized into distinct, specialized, functional zones. We start just beneath the collagen capsule with the subcapsular sinus, which is where the lymph first drains in from the afferent lymphatics.
Deeper inside, we find the cortex. This is predominantly the B cell area. It often contains these highly active structures called follicles or germinal centers where B cells are proliferating.
And beneath that?
Beneath that is the paracortex, which is the high-traffic T cell area. And then finally, in the center, you have the medulla, which is the central core. It contains streams of antibody-producing plasma cells and macrophages.
The circulation within the system isn't a simple flow path, is it? It's a highly dynamic cycle. How do the lymphocytes actually enter the node from the blood in the first place?
They enter via these very specialized structures called high endothelial venules, or HEVs, which are primarily located in that T cell paracortex. These venules have unique surface markers that allow circulating lymphocytes to grab on and actively migrate out of the blood and into lymphoid tissue.
And then they just browse.
They browse. They circulate through the node, scanning for their specific antigen. If they don't find it after a few hours, they exit via the efferent lymphatics to continue patrolling elsewhere. This Constant recirculation is absolutely non-negotiable for effective immune surveillance.
Then we have MALT, which stands for mucosa-associated lymphoid tissue. This sounds like a more decentralized defense system.
It is. And MALT is crucial because it protects the massive surface area of our mucosal boundaries, the gut, the respiratory tract, the urogenital system, which are constantly exposed to external pathogens. It's a diffuse collection of lymphoid tissue, including the tonsils, Peyer's patches in the small intestine, and other lymphoid follicles.
So its function is highly specialized for local defense.
Correct. Lymphocytes in MALT are primed locally by antigens that pass through the mucosal barrier. Once they're activated, they migrate to effector sites, like the lamina propria, which is the layer of tissue just beneath the epithelium, to carry out defensive functions right at the source of entry. It's the ultimate perimeter defense system.
Now, for the key players in adaptive immunity, the lymphocytes. They make up 20 to 40% of the white blood cells, and they're the smallest leukocytes. They look deceptively simple under a microscope, mostly just a big nucleus with a thin rim of cytoplasm.
But looks are incredibly deceiving. They are a highly heterogeneous collection of cells with lifespans that range from short-lived effector cells, which last only days, up to months or even years, for the memory cells that grant us long-term immunity.
And their constant dynamic movement, that recirculation between blood, lymph, and tissues, is the only way the immune system can effectively sample the entire body for threats.
It is. A lymphocyte might be in your blood 1 minute and in a lymph node in your armpit the next.
Let's trace their genesis, starting with B-cell development, which happens in the bone marrow.
Right. So hematopoietic stem cells mature into progenitor cells, and the critical step here is a process called VDJ recombination. This is the rearrangement of the immunoglobulin, or IGLI, genes.
A genetic shuffling.
A random genetic shuffling. It allows each individual B cell to create a completely unique surface receptor. This means that the total B cell population, theoretically, can recognize almost any antigen imaginable. This is the source of our vast immunological diversity.
But diversity breeds risk. How does the body ensure that this random system doesn't produce cells that will attack the host's own tissues?
That's the job of negative selection. Any B cells that have high-affinity receptors for self-antigens that are present in the bone marrow environment are recognized and eliminated, usually through apoptosis or programmed cell death.
And our sources say that about 25% of B cells are eliminated during this process.
It's a significant cull. It's a necessary initial safety check. The survivors then leave the bone marrow expressing surface IgM and IgD, ready to go out and patrol.
The T-cell development pathway is even more rigorous, and it takes place entirely in a different organ, the thymus.
It's much more rigorous. T cells start as progenitors that migrate from the bone marrow to the thymus. And the thymus is where they undergo this intense two-step selection process designed for dual assurance. They have to be functionally competent, and they must be non-reactive to self.
The first step occurs in the cortex of the thymus, and it's a brutal entry test.
It is. In the cortex, the T-cell receptor, or TCR, gene rearrangement occurs, and the developing cells express both CD4 and CD8 markers. Then comes positive selection. Only T cells that can successfully recognize self-MHC molecules, the body's own identity tags, are allowed to survive.
So if a T cell can't see the self-MHC tag, it's useless and it dies.
It's useless and it dies. This stage is incredibly selective. Our sources note that a mere 2% of T cells survive this first stage.
Wait, a 2% survival rate? That is an astounding level of what seems like waste, but it must be efficiency. Why is the body so incredibly strict about keeping only the T cells that recognize self?
Because the T-cell receptor is structurally incapable of recognizing an antigen unless that antigen is presented by an MHC molecule. So if the cell can't recognize the body's own MHC framework, it will never be able to recognize a pathogen that's being presented by that framework later on.
It would be like having a security guard who can't read the blueprints of the facility he's meant to protect.
That's a perfect way to think about it. That puts the 98% attrition rate into perspective. But then, the survivors move to the medulla for the second, even harder test.
And that is?
Yes, the final, crucial step, negative selection. This stage eliminates any of the T cells that survived positive selection but react too strongly to self-antigens that are presented by specialized unique APCs. This is the ultimate self-tolerance check, eliminating the risk of autoimmunity.
And this phase is even more brutal.
It is. It eliminates up to 95% of the remaining T cells.
That's almost universal elimination. It ensures that the very few T cells that survive and leave the thymus are functional, highly specialized, and absolutely self-tolerant.
It is the ultimate quality control process in biology, I would argue. The survivors leave the thymus as naive, mature T cells, either CD4+, which become helper T cells, or CD8+, which become cytotoxic T cells, ready for circulation.
Let's break down those T cell subsets. First, the helper T cells, TH cells, the CD4 plus cells. They are the orchestrators, the command center of the adaptive response.
TH cells are activated when they encounter an antigen that's being presented by a professional APC, like a dendritic cell, on an MHC class II molecule. The CD4 co-receptor on the T cell physically binds to the MHC-TI molecule, ensuring the right connection.
And once activated,
They proliferate and begin secreting a massive array of cytokines chemical signals to coordinate the entire immune response. They're the generals.
and we have to distinguish between the major functional subtypes of these helpers.
We do, because they control the direction of the immune response. Th1 cells primarily secrete interleukin-2, IL-2, and interferon gamma, IFN gamma. These cytokines promote cell-mediated immunity. They stimulate cytotoxic T cells and macrophages to handle intracellular pathogens like viruses and certain bacteria.
And Th2 cells swing the response in a different direction toward humoral immunity.
They do. They secrete IL-4 and IL-5, which are specialized in stimulating B-cell activation, promoting IgE production, and recruiting eosinophils. They are critical for handling extracellular pathogens, particularly parasites, and they are the key drivers of allergic disease.
Next, the ultimate executioners. Cytotoxic T cells, CTLs, which are CD8 plus cell. These are the kill cells responsible for direct cellular elimination.
They're activated when they recognize an antigen presented on MHC class I molecules. And MHC class I is expressed on virtually all nucleated cells in the body. This means CTLs can identify and target any cell that has been compromised, whether it's virus-infected, cancerous, or a foreign transplant cell.
And the CD8 co-receptor ensures the correct binding to MHCI. Their killing mechanism is fascinating because it's so clean. They kill by inducing apoptosis or programmed cell suicide.
Yes, they employ two primary weapons, perforin and granzymes. Perforin is released, and it polymerizes in the target cell's membrane, creating literal pores or holes. Granzymes, which are a family of potent serine proteases, then enter the cell through these pores and activate the cell's own internal apoptotic cascade.
So the target cell kills itself neatly.
Exactly. It breaks down into small membrane-bound packets that can be safely cleaned up by macrophages. This prevents the dangerous release of cellular contents and widespread inflammation. It's a very tidy assassination.
Finally, the critical component of immune self-discipline, regulatory T cells, or TREGs. They express CD4 plus and the IL-2 receptor CD25 plus CELI5 era. They're the essential immune brakes.
They are crucial for maintaining self-tolerance and preventing the rest of the immune system from overreacting and causing damage. They suppress the activation and proliferation of other T cells, both through direct contact and by secreting inhibitory cytokines like interleukin-10, IL-10, and transforming growth factor beta, TGF-beta.
So their function is vital in preventing autoimmune diseases.
And in promoting tolerance. For instance, in the gut, they're responsible for oral tolerance, so we don't react to food proteins. And they're critical during pregnancy to tolerate the fetus.
Wrapping up the adaptive cells, let's look at B cells and the product they are famous for. Antibodies, or immunoglobulins, Igs, the foundation of humoral immunity.
Right. Once a B cell is fully activated, usually with help from a helper T cell and its specific antigen, it differentiates into a plasma cell, and this is a dramatic morphological change.
The plasma cell is purely a factory.
Absolutely. Structurally, it is characterized by massive amounts of rough endoplasmic reticulum and a large eccentric nucleus. That RER is necessary for the rapid synthesis and secretion of hundreds or thousands of antibodies per second. It is a cell that is completely dedicated to protein production.
And the antibody structure is versatile, categorized into 5 main types: IgM, IgG, IgA, IgE, and IgD, each with a distinct functional role.
IgE is the powerhouse. It's the most abundant immunoglobulin in the serum, and critically, it is the only class that can cross the placenta. This provides immediate passive immunity to the fetus and newborn, offering protection until their own adaptive immune system is fully functional.
And IgA is the mucosal champion.
That's a great name for it. IgA is secreted in breast milk, saliva, tears, and onto all mucosal surfaces.
Yeah.
It often exists as a dimer, 2 units linked together. It is the first line of defense at the boundary between the body and the external environment, preventing pathogens from adhering to and crossing our epithelial surfaces.
What about IgM?
IgM is the primary antibody in the initial immune response. It's often found as a pentamer structure, 5 units linked together, which makes it highly efficient at binding multiple antigens simultaneously and kicking off the response.
And antibodies don't just neutralize pathogens directly by binding to them, they can also trigger a massive amplification system.
Yes, they initiate the complement cascade. When two or more antibody molecules, usually IgG or IgM, bind to an antigen on a cell surface, they activate what's called the classical pathway of the complement cascade. This is a sequence of plasma protein activations that ultimately leads to inflammation, opsonization tagging targets for phagocytes, and the formation of the membrane attack complex.
Which does what its name implies.
It directly punches holes in the target cell membrane and causes it to lyse or burst.
Now that we understand the complexity and specialized jobs of these billions of cells, we have to look at the continuous process that produces them all. Haemopoiesis. This is an industrial-scale operation operating with phenomenal speed and precision.
And the location of this factory shifts dramatically throughout development, which demonstrates the body's adaptability. It actually begins in the yolk sac in the early fetus. Then it transitions to the liver and spleen during mid-gestation, but by the 6th month, the bone marrow takes over as the primary and permanent site.
In adults, haemopoiesis is confined primarily to the core skeleton, the flat bones, so the vertebrae, sternum, ribs, pelvis, skull, and the ends of the long bones.
And this site is also highly dynamic. We distinguish between red marrow, which is the actively hemopoietic cell-generating tissue, and yellow marrow, which is largely inactive and consists mostly of fat and connective tissue.
But the yellow marrow is a critical reserve.
It is. In cases of severe hemorrhage or pathological demand, it retains the potential to rapidly revert back to active red marrow to ramp up production.
The bone marrow is more than just a collection of cells. It's a carefully structured environment, a biological niche that provides all the necessary support and regulatory signals.
Right. The stroma is that supporting framework. It's composed of endothelial cells lining the specialized sinusoidal capillaries, reticulum cells, which provide the supportive architecture and, most crucially, secrete essential growth factors, chemokines and neuropeptides that dictate stem cell fate, and specialized fibroblasts that produce the reticular fibers, the type 3 collagen, that form the physical scaffold.
So the stroma acts as the physical scaffold, the HR department by secreting growth factors, and the quality control barrier.
Precisely. Those sinusoids, the wide, thin-walled capillaries, form a very tight barrier. Mature blood cells must actively squeeze and pass through the endothelial cells themselves to enter the general circulation. This filtration ensures that only fully differentiated mature cells are released.
It prevents the premature entry of precursors.
Exactly.
At the absolute top of the system are the hematopoietic stem cells, or HSCs. These are the true masters of the factory floor, defining the capacity of the entire system.
And they are exceedingly rare. They make up maybe 0.05% of the total marrow cells. But HSCs possess two incredible, almost magical properties. Self-renewal, they can copy themselves to maintain the stem cell pool throughout a person's life, and multipotency, they can generate all the different blood cell lines.
The logistical challenge here is staggering. How does the system maintain the balance between making copies of itself and differentiating into other cells?
It's dictated by the microenvironment of the niche and very specific chemical signals. Only a tiny critical number of these cells are needed to completely repopulate the entire bone marrow after trauma or clinical interventions like chemotherapy. This highlights their intense potency and self-sufficiency. Clinically, they're often identified by the surface marker CD34+ pin.
HSCs then commit. They differentiate into committed progenitors. These are cells that have lost the capacity for self-renewal but have committed to a specific lineage, responding only to very targeted growth factors and hormones.
Right. Examples include the common myeloid progenitor, which leads to the granulocytes, monocytes, erythrocytes, and platelets, and the common lymphoid progenitor, which leads to B and T lymphocytes and NK cells. Scientists define these lineage commitments by their ability to form specific colonies in vitro, which is where you get names like CFU-GM, which stands for colony-forming unit granulocyte monocyte.
Let's follow the first lineage, the erythrocyte lineage, or erythropoiesis, responsible for those 6 million cells produced every single second.
The whole process is controlled primarily by the hormone erythropoietin, or EPO, which is produced by the kidneys in response to tissue hypoxia or low oxygen. EPO is the hormone that acts as the emergency production signal, and it specifically stimulates the earliest committed red cell precursors, the BSUE cells.
The maturation is incredibly fast, only 5 to 9 days from start to finish. What are the key milestones in that process?
It starts with the proerythroblast, which is a large, very blue staining cell because of all the ribosomes making protein. This cell undergoes rapid division. This moves to the erythroblast stage, where division continues, and critically, hemoglobin synthesis starts in earnest.
Next is the normoblast stage. This is the visual precursor to the mature cell.
It is. In the normoblast, the nucleus starts to shrink and condense. It becomes pinodic, a dense, dark, dying mass, as the cell optimizes all of its internal space for HB storage. The climax of this process is when the nucleus is actively extruded, just pushed out of the cell.
And that transforms it into a reticulocyte.
Correct. The reticulocyte is the last precursor stage before full maturity, and its count is often used clinically.
Why is that?
Because the reticulocyte still retains some residual cytoplasmic RNA, which gives it a slightly bluish tint, but it has no nucleus. It is released into the circulation, and it matures into a fully functional erythrocyte within a day or two. So the reticulocyte count is the immediate, real-time indicator of the bone marrow's current rate of red cell production. If someone is anemic, you look at the red count to see if the marrow is mounting an appropriate response.
Moving to the granulocyte lineage, or granulopoiesis, this process is focused on internal organization and nuclear changes, starting with committed progenitors like CFU-GM.
The morphological changes are very distinct and sequential. It starts with the round nucleus of the myeloblast, then moves to the formation of the first primary granules in the promyelocyte. Then the appearance of the secondary, specific granules in the myelocyte. Then the nucleus starts to indent, becoming kidney-shaped in the metamyelocyte.
And then we get the signature look of the mature neutrophil.
Yes. The nucleus develops the characteristic C shape of the band form. And finally, the segmented, multi-lobed structure of the mature neutrophil. And maturation takes approximately 12 days, significantly longer than red cells.
And unlike the quick release of reticulocytes, these cells are stored, creating a reserve force.
They are. They're stored in the marrow for up to 6 days before release. And this storage pool is crucial. In cases of acute bacterial infection, the bone marrow can rapidly mobilize this reserve, causing a sudden spike in circulating neutrophils. This is often indicated by the presence of those immature band forms in a blood smear, which reflects a high-demand factory output.
Finally, the monocyte and platelet lineages. Monocytes are also derived from that CFU-GM progenitor.
Monoblasts mature into monocytes, which quickly leave the bone marrow. As we discussed, they don't linger in the blood, but migrate into tissues to differentiate into their specific, highly versatile forms, macrophages, osteoclasts, or dendritic cells like Langerhans cells.
And the platelet lineage, thrombopoiesis, is the most visually bizarre lineage, resulting in the megakaryocyte.
It's completely unique because it results from a process called endomitosis. The megakaryoblast replicates its DNA over and over without the cell ever dividing. This creates a giant polyploid cell with a massive multi-lobed nucleus. This giant cell is purely a platelet manufacturing hub.
And the final act of the megakaryocyte is literally to fragment itself.
Correct. It extends these large cytoplasmic pseudopods, called proplatelets, right through the endothelial lining of the sinusoids. These long strands are exposed to the flowing blood in the sinusoid, which shears or fragments them off, releasing the platelets directly into the circulation. It's an active mechanical process of shedding its own cytoplasm to create the clotting crew.
We have completed an exhaustive deep dive, moving from the macrophysical properties of a single blood drop all the way down to the cellular architecture and the industrial logistics of the bone marrow.
We've covered everything. I mean, the necessary density of blood, that viscosity of 4.75, the ingenious engineering of the biconcave erythrocyte, the specialized weaponry of the granulocytes.
And the essential, brutal efficiency of T-cell selection in the thymus, where 95% of candidates are discarded just to ensure self-tolerance.
Right. What's fascinating here is the sheer interconnected complexity of it all. The entire system is dictated by these constant feedback loops, from the EPO produced by the kidney, telling the bone marrow what to build, to the constant recycling of 6 million red cells every single second.
How a single hematopoietic stem cell dictates the entire complexity of oxygen transport, immune defense, and vascular repair, all requiring this constant, perfectly regulated balance.
This knowledge is foundational. For you, the learner, understanding these processes provides immediate, tangible context for concepts that affect everyone. Why a simple complete blood count, a CBC, is so incredibly informative, what is actually happening during an allergic reaction, and the fundamental pathology behind chronic inflammation or various forms of anemia.
So if we connect this to the bigger picture, given the incredible logistical precision and the sheer quantity of cells produced every single second, what happens when the factory floor in the bone marrow faces dual immediate stress, for instance, simultaneously dealing with acute blood loss from a hemorrhage and a massive bacterial infection requiring a neutrophil launch?
The factory must pivot, and pivot instantly. How quickly can the body truly switch from maximum red cell production, which takes time and a lot of iron, to launching a full-scale white blood cell defense, which demands a completely different set of progenitors and growth factors? That rapid regulated pivot, shifting the entire hemopoietic focus in real time, is one of the most remarkable and life-dictating functions of the human body.